Assalamualaikum and salam sejahtera. I'm Akmal bin Rahim, Science Officer Entomologist. I will present my article entitled Key Breeding Containers of Dengue Vectors in Outbreak Localities in Federal Territory of Kuala Lumpur. For introduction, dengue is a mosquito-borne disease that rapidly spreads throughout tropical and subtropical regions. An estimate 2.5 billion people live in dengue-endemic countries and millions of cases occur annually. Dengue has become a major public health issue in Malaysia with an increasing trend of dengue outbreak every year. The number of reported dengue cases remain above 100,000 cases from 2014 to 2016. Meanwhile, in Federal Territory of Kuala Lumpur, the number of reported dengue cases remain above 8,000 cases from 2016 to 2017. Aedes aegypti and Aedes albopictus are known as dengue vectors in Malaysia. Their effectiveness in transmitting dengue is based on their ability to breed in water-holding containers close to humans and their eggs' ability to survive for months in dry conditions. Identifying Aedes key breeding containers are an important key to preventing dengue epidemics. In this study, we aim to identify the key breeding containers of Aedes mosquito and to evaluate the actual risk of dengue transmission in the dengue outbreak localities. For methodology, Kepong District was selected as the study site. A house-to-house survey was conducted at the 38 outbreak localities from January 2016 to December 2017. All water-holding containers were inspected for the presence of mosquito larvae around 100-meter radius from the case index. The potential and positive mosquito breeding containers were counted and recorded. All collected mosquito larvae were transported to the laboratory for species identification. The species of mosquito were counted and recorded. Larva indices used were house index, container index, and Butu index. The threshold value for larva indices are as follows. House index low than 1%, container index below than 10%, and Butu index below than 5%. Data were compiled into a Microsoft Excel. For the result, a total of 2,067 water-holding containers were inspected, of which 227 containers were positive for Aedes mosquito larvae. From these positive containers, 72 were plastic containers, and followed by water drum, drain, flower pot, toilet, flush tanks, tire, and gully traps. A total of 2,118 mosquito larvae were collected and identified, and of these, 1,176 were Aedes aegypti and 942 were Aedes albopictus. Aedes aegypti bred in all types of water-holding containers found in that study site. Overall, 959 houses were surveyed, and of these, 116 houses were found positive for Aedes mosquito larvae. The result of larva indices were ranged between 0 and 52.94% for house index, between 1.72% and 32.76% for container index, and between 2.33 and 75 for Butu index. The average of larva indices at the study site were found higher than the threshold index, with house index 5.1%, container index 10.98%, and Butu index 23.67. For discussion, Aedes mosquito larvae preferred to breed in plastic containers, water drums, drains, and flower pots. Most of these containers were located outside the houses and were also uncovered. The uncovered water-holding containers for a long period of time will be a high potential for mosquito breeding habitats. The larva indices found in this survey were high, especially in some outbreak localities. This indicates that the high risk of dengue transmission is still occurring. The evidence-based findings from this study were useful in providing health education to local communities on the importance of search and destroy the water-holding containers surrounding their houses, and these preventive activities should be carried out weekly. The systematic planning by the health department focusing on eliminating high potential breeding containers in the outbreak localities will make the vector control activities implemented effectively, efficiently, and cost-effective. This is our references. Until then, thank you. Aduh.